It's a great pleasure to be here for the first time in Brazil. I'd like to tell you a brief overview about basically everything I've done since I started my PhD. And it's all turns around the question of forces and stresses in living matter. So to give you an idea, one of the key characteristics of life, of living matter, is that it's able to generate internally, at the nanometer scale, the forces that it's going to use to move, to change shape, and to exert its function. And so these forces originate from the action of individual proteins called molecular motors that consume ATP and exert these forces at the molecular scale, which are then transmitted to intracellular fluctuations, cell motion, and even whole animal development. I don't know what that video is not playing. So I'd like to tell you a little bit about the two different things that these forces can do in living matter. The first one is to be used at a given scale to produce motion. The second one is to be transmitted to larger scales to produce stresses that can then be utilized collectively at, to generate larger scale motion. So that's going to be the two parts of my talk that are essentially independent. The first one is about force transmission and stress transmission through biological matter. And the second part is about forces going back down to the Brownian scale, forces as they emerge from Brownian noise and from thermal fluctuations. And that part has actually broader implications than just biological forces. In both of these cases, I'm going to make theories for how these forces emerge and transmit. But these theories are pretty hard to confirm experimentally. And one reason for that is that you cannot, by eye, see forces or see stresses. And so in both cases, I'm also going to spend some time telling you about methods I've developed to infer stress fields and force fields from experimental data, and so to be able to directly confirm these predictions from experiments.
You're making assumptions and then try to prove, or you're calling theory here?
I'm going to make predictions connecting local assumptions to global forces, for instance. This is going to be much clearer in a few minutes. By the way, don't hesitate to ask any questions. Otherwise, my talk will be too short. So moving on to the first part, which is about force transmission in biological networks. So it's really a biomechanics problem, asking how mechanical forces are transmitted inside living matter. It's a work that I've done with my PhD advisor, Martin Lenz, and continued with a professor at Munich University. So these two are theorists, and then it's complemented by an experimental part by the group of Mingo at MIT. And I'd like to motivate it to start with an everyday life example, which is what happens when you scratch your skin, start bleeding, and have an open wound. What's going to happen then is that your body is going to secrete a blood clot that is going to enter the wound and start filling it. And that blood clot is going to be contracted by the effect of blood platelets, the same platelets that you give when you give blood. And so the blood clot is made of an entangled meshwork of hundreds and hundreds of many filaments. And the platelets are going to invade this network, start pulling around, exert forces, and contract the clot. And that's what closes the wound and leads to the stop of bleeding. And that's the first stage of wound healing. The reason why these platelets are able to generate forces is because they have, as an internal structure, another meshwork of entangled protein filament called the actin cortex. That's part of the cytoskeleton, the skeleton of the cell. And that actin cortex is put under tension actively by the action of myosin filaments. Myosin filaments are made of hundreds of myosin heads, which are individual motor proteins, which perform elementary force generation steps called power strokes. So a myosin motor grabs an actin filament. Then there's a sugar molecule, an ATP molecule that comes around. It hydrolyzes it and performs a single step forward, always in the same direction. And that's by harnessing these molecular level forces and transmitting them all the way to the centimeter scale, that you're able to close the wound and stop the bleeding. So that's a nice biological scenario, and I think there's a question that a theoretical physicist can address that occurs at 7 different scales in this problem, which is the way that a fiber network transmits active forces from the local scale to the global scale, both in the atomizing cortex with the action of myosin motors in actin filaments, and in the blood clot at a significantly larger scale, where the action of platelets on the fibrin filaments results in closure of the wound. So the question I'm going to ask is, given the action of these local force exerters inside the network, what is the resulting large-scale stresses? How do these forces transmit? And how do you efficiently produce large-scale stresses? How do you make an efficient stress-generating material? So the first player here is going to be the active units. And I'm going to take for granted their properties. Their properties are that they have a certain shape. They're characterized by the size, R naught. They exert a certain force, F, and they come at a certain number density in the gel that I'm going to call rho. And really, I've defined force on size here pretty loosely, but the quantity that is well defined and that quantifies well the action of an active unit like this is the force dipole. Because these proteins or these cells are not like stuck on a substrate. They're free-floating in the gel, and so they cannot exert a net force on average. So what they can exert is a net force dipole, in which they exert one force at a point and a counter force at the other point. And if the force dipole is negative, meaning that the forces are anti-aligned with the vector, position vector, then you have a contractile dipole with a negative sign. And so I'm going to try to make a theory relating this. Yes.
The dipole is the minimum you need, right? You could have quadrupoles and stuff like that.
Sure.
Lisa Manning, I think she puts quadrupoles here, doesn't she, when she does the stretch unit?
So Lisa Manning works with, I think it's dipoles.
It's only dipoles? Anyway, dipoles are enough. Let's continue.
Yes. I think it's dipoles. The dipoles are the ones that are going to get to macroscopic stress to emerge. I'm going to show that in a second. So the goal of this first part of the talk is going to establish a theory that relates these properties of the active units and the network properties to the macroscopic stress that you get in the network. And for that, we have an exact result that can help us connect the far field forces, the boundary force dipole, for instance, to the local forces exerted by the active units. This exact result is called the mean stress theorem. It does not make any assumption other than being static, so at static mechanical equilibrium. And what it says is that the macroscopic coarse grain stress that you get by considering this whole unit as a small elastic piece is equals to 1 over the volume, or the surface if you're in 2D, but I'm generally going to talk about 3D networks here, times a difference of 2 terms. One is minus the sum of the local dipoles in the network, so the dipoles exerted actively by your force-generating units. And that's a given of the theory. But the other term is the integral of the stress along the network. So here I've written it all for the trace of the stress tensor, but you can do everything again with tensorial indices, which is going to give you the full anisotropy of the theory. So that integral of the stress here is the way, at equilibrium, the forces transmit through the network. And this is a beautiful result, I think. It's exact. And at the same time, it's somewhat useless, as it is so far, because there are 2 unknowns. You don't know the macroscopic stress. That's what you want to guess. But you don't know either the integral of the stress through the medium. However, there's one precise context in which this theorem can be very useful, which is the case of a linear elastic material. if you're in a linear elastic material, meaning that the displacement at all points in response to a point force are proportional to the force, then there's kind of a miracle that occurs. And the average stress here, the integral of the stress vanishes exactly. That's true in a linear homogeneous medium. It's also true in a disordered medium with homogeneous disorder, so on average homogeneous, which is far less trivial. And what it means is that you have an exact relation connecting the macroscopic stress to the local force dipole density. And that relation can be read as the large-scale stress equals the density of active units times their force times their size, such that the force times R naught is the dipole that they exert.
Sorry, I'm confused about vectors versus just numbers. So capital F is just a number, it's not a vector, right?
So here, capital F is a number, and R naught is a number, such that the product of the two is the force dipole.
So can you just remind me, what's the definition of DI?
The local force dipole is the sum for one given active units of the force times the position over all points at which the dipole exerts forces, the unit exerts forces. So the total force vanishes, so that makes it independent of the starting point for the positions.
Okay. Thank you.
Good. Any other questions so far? So this is an exact result. It predicts what the macroscopic stress, or the trace of the stress, it's the contractile or expansile part of the stress, is given the density of active units, their force, and their size.
I label them? So
sorry, that's for all dipoles in the gel. So if I get back to the previous, that would be a sum running over all these red units here, exerting forces.
But what does J, what's the difference between I and J? So J is a sum already.
J, that's within I. J is...
I see, okay. So you divide it into little subsections?
Yes,
okay.
yes. This whole result is independent on the way you divide things.
Okay. And sigma can be positive or negative, I guess?
So sigma, if it's positive, it means that you're under tension, and if it's negative, you're under compression.
Okay.
You're pushing underwater. And that's why there's a negative sign between the dipole and the dipole and the stress. The stress is minus the pressure in that context. So this is a nice result. It's exact, and I think one of the striking features that it reveals is that it does not involve at all the network properties. I have not at a single point introduced the elasticity or the Poisson ratio of the network. I've only said that it's there and it's transmitting forces. And on the other hand, it's crucially dependent on the geometry of these active units, of these active force exerters, through the fact that the dipole is the quantity that matters. However, so far I've made a key assumption, the fact that the network responds linearly to any forces, and that assumption turns out to be wrong. If you look at the different nonlinear mechanisms that you have in such networks, one of them is what I call plucking. It's kind of a fun one. It means that if you're exerting a force that is perpendicular to a filament, it will put that filament under tension no matter what the initial direction of the force was. And so that's going to bias everything towards tension, but it's a somewhat local effect, and I'm not going to consider it too much here. The other nonlinear effect that I want to consider a lot here is buckling. If you pull on a filament, it's going to resist and transmit forces, while if you start pushing on it, initially it will respond linearly if the forces is very low, but it's at force levels relevant for biology, they will actually tend to buckle, yield, and to stop transmitting forces under compression. And if you include that element in the force transmission theories, you get a completely different result. Indeed, if I start, so this is a simulation I've made where each of the, so it's a regular network made of springs, where each of the springs is made of 2 hinged springs. Each of the edges is made of 2 hinged springs that can individually buckle. And so under compression, each of these lattice edges will yield and stop transmitting compressive forces. I'm now going to slowly increase the force and go from this linear force transmission, in which I've put an extensile force dipole here that pushes around and transmits extensile forces on the boundary, as predicted. And I'm going to go through the buckling threshold, and we'll see what happens. So you see that stress accumulates, buckling occurs here, and then there's something really striking that happens. You had initially an extensile force dipole that was pushing around, and it was pushing on the boundaries. But at large enough forces, that's no longer what's happening. What you have is contractile forces in the perpendicular direction that emerge and start becoming increasingly contractile. So you have a rectification of the forces towards contractile forces. That's going to happen no matter what you do. Essentially, if you start pushing, you're going to have transverse tension. If you start pulling, you're going to have radial tension. And there's no way around that. And if you keep increasing the force, actually you will even lose the information about the orientation of the initial dipole. And you will get essentially isotropic contraction. So that's what you would get at very large forces exerting an extensile force dipole. On the boundary, you see that all arrows point towards the center. So that's kind of a striking effect. Forces are rectified and transformed into isotropic contraction because of buckling. It works also in 3D. It's essentially something you cannot avoid because only the tensile forces are transmitted and the compressive ones do not transmit. If I want to grossly summarize this, before, this was the part that matters, the local force dipoles. That was during when the response was linear. Now, actually, you can completely neglect the local force dipoles. And what matters is the integral of the stress, the way the forces are transmitted and select contraction through the network. And so now we've moved to a scenario where geometry does not really matter for force transmission. Only the magnitude of the forces matters. So now I'm going to go to a slightly larger scale and ask how these forces transmit towards the far field. Are there any questions so far? So let's look around. I've said that everything becomes contractile, so now I'm only going to consider contractile units. And I'm going to look around these contractile units that pulls isotropically on the network. And it transmits these blue tensile forces in the radial direction. But these are partially compensated by the red compressive forces in the
You're assuming the motors are acting everywhere?
So now I'm looking at one single motor isotropically contracting. Because in the previous part, I've shown that this is, in the far field, this is the effect of a motor. So at the linear level, you have compressive stresses that partially compensate the tensile stresses in and that prevent them from propagating. And that leads to a stress decay when going away from the active unit that goes as 1 over r to the d, so 1 over r cubed in 3D. D is the space dimension. And that's the linear elasticity result. Now, if you add buckling to the story, so we've just seen that it changes a lot force transmission. Now, all the compressed ladder steps here start to wiggle out, and they cannot transmit forces anymore. So all forces is carried by tension. And so if you look at force balance over a small portion of the spherical shell, the inner force and the outer force have to compensate. And so the total stress has to decay as 1 over r to the d minus 1, so that it's conserved over its spherical shell. And so that's what I call rope network force transmission. It's how forces transmit in material that cannot withstand any compressive forces. And it's longer ranged than linear elasticity. So it leads to an amplification of the forces when going away. Yes. Here, that's force. Yes. If you're under compression. If you're under tension, you resist. If you're under compression, you buckle. So that was what happens for a single active unit. Now what happens for the stress generated collectively by many active units contracting? So now I'm going to start with this linear scale, linear level simulation in which you have many different units, and I'm going to increase the force slowly. And what you've seen now is that each of these active units surrounds itself by a buckled zone, a region in which filaments align, transmit radially the forces, and there's no relevant compression there. And then in the far field only, like far away from each of these active units, you get back to linear force transmission because you're not strong enough to buckle. So this range of buckling increases with increasing force. It actually scales as the force over the buckling force to the power 1 over d minus 1. So in 3D, it scales as the square root of the force of these active units. And that's the region in which each of these active units amplify its forces. So in terms of global force transmission, it's a global stress generation. It's a bit like replacing each of these active force exerters with a larger unit that exerts the forces linearly, but at a longer range, because the forces was transmitted, was conserved in the rope network region. And so we get back the same macroscopic stress equation as before, except that now it's not the size of the active units that matters. So it's density of active units times their force times the effective size, the range at which they're able to buckle. And that increases with the force, so that leads to a stress that increases non-linearly with increasing force of the active units. So that's how it goes, provided that these regions do not start overlapping. If they start overlapping, you have to have something else that happens, because a filament can buckle only once. And so let's look at what happens in the strongly nonlinear regime when you go to larger forces and all the filaments buckle. So what you see now is that the morphology of the network kind of self-organizes into a pattern that does not change much anymore with increasing force. Each of these filaments tenses between nearby active units, and it's like active units playing a tug of war with each other. Each of them pulls, and there's a balance that's obtained this way. And you can see that essentially, if I plot the Voronoi cell of each of these around each of these active units, there's force balance, there's tug of war force balance at each of these of the boundary. So this size is now the new effective size of our active units. So if I now try to make a phenomenological theory for prediction for how much stress you would get in that network, you would replace the size of the active units with this new effective size, which is the distance between them. So that's the density to the power minus 1 over d, 1 over the density to the 1/3 in 3D. Yes?
How do you initialize the active units?
Here,
random? Or?
They're just randomly positioned. So the active units are initially randomly positioned. And here, because it's prettier, I've shown you a network where I've randomly cut bonds too, and that is pretty soft initially. That's thought to be a relatively good model for these networks. But you would get exactly the same with regular spacing, regular network.
My question is, so you start them randomly, once the forces balance each other, do you get some kind of regularity out of the structure, or they keep the same? You know what I mean? Like,
I think it's closer to being an irregularity that emerges. They tend to cluster and to attract. That's not something I've looked too much into.
Thank you.
But quite often, they would collide. Yes, because they're sucking in. They're little black holes that suck in all the network around, and so they tend to collide if the forces are too large. So now you have a macroscopic that's predicted as being the density of active units times their force times the distance between them. So that's proportional to the density of active units to the power minus 1 over d. So you're essentially in a strongly interacting regime in which you're back to a linear behavior in the force, but the behavior is nonlinear in the density of active units. Adding an active unit has a decreasing effect. The sparser they are, the more room they have for amplification, and so the stronger they are. I can summarize all of these different behaviors on a single phase diagram in the plane of logarithm of the active unit density, rho, and log force. If the force is too low, the effective size of the active units is, well, their real size, R naught. If you start increasing the force, you start being controlled by the force that they exert, where the range at which they they can buckle dominates the force generation. And if you keep increasing the force, you reach that density-controlled regime in which you self-organize into a strongly interacting regime. So that gives both a set of quantitative predictions for the level of stress given the properties of these active units and the properties of the network. One key prediction being that there's long-range tension as 1 over RD to the d-1 exerted by each of these active units. But there's also kind of a strategical prediction that it gives for living systems, which is that if you make active units sparser and stronger, so if you cluster them, they're going to be able to buckle more and to buckle further because they're less hindered by their neighbors. And so clustering leads to larger stresses. So that's a prediction of this theory. The stronger and sparser your active units, the more efficient you will be at generating stresses. Now, how does this work in real life? So far, I've told you only about theoretical predictions. Let's move back to experiments. And I've told you about the stress in a blood clot. And if you measure the experimental stress in such networks, it's of the order of 100 pascals. And that's what you would obtain if you just put one between two rheometer plates and measured how much it pulls between the rheometer plates. If you now start looking at what the linear elasticity theory predicts, you get something of the order of 9 pascals. Because these platelets are 2 microns, they're pretty small, they exert large forces, and they're pretty far apart. They're pretty sparse. So that's not in the right ballpark. There's more than an order of magnitude of discrepancy between the two. But now if you put in numbers, seeing that these forces are large, 15 nanonewtons are large biological forces, they're able to buckle at a virtually infinite range, hundreds of microns. And so really, what I predict with my theory is that you're deep into this density-controlled regime in which what matters is not their size, but the distance between them. And so my prediction is of the order of 70 pascals, which is well within the error bars for these biological numbers. So it seems like this is the right way to think about cells generating stresses in an extracellular matrix. There's also interesting predictions that have not been tested yet, such as the fact that if you changed the concentration of platelets, because of this strongly interacting nonlinear behavior, the stress would vary as the power 2/3 of the density of platelets, so in a nonlinear way. Yes?
How do you do this measurement with experiments?
This one?
Yeah.
This one was done in the 80s, and they really put a network between 2 plates and looked at the traction force. So they polymerized a reconstituted clot, and they measured actual stresses. I have not found more recent data on doing that exact same experiment. So that's experimental hints that my prediction for the nonlinear force transmission is valid at the collective scale. Recently, I've also teamed up with an experimental group, the group of Minguo at MIT, who's looking at force transmission around single cells. So this is a unique contractile cancer cell. It's sitting in a 3-dimensional collagen network, and it's pulling at both of its ends. You can almost see alignment of the fibers generated by these stresses. And so this was a good candidate to test this 1/r² stress transmission that I've predicted. But the problem is that measuring these local stresses is highly challenging. what people generally do for macroscopic stresses is to use either a rheometer or to look at traction force microscopy looking at displacements at the boundaries. So that works well. It's possible to extend these methods to infer stresses in the bulk, but you need to assume that the network is homogeneous and linear, and that's not at all the case here. So what we've proposed is a new method to do that, which uses the nonlinear properties of the network. So these networks, when you start pulling on them, they stiffen and stiffen before they break. And so that gives us a way to pull back the stress from a measurement of the stiffness. So the way it works in practice is that you use optical tweezers. So you use a laser to move beads through the network and measure the force displacement relations. So that network is infused with beads. And using these force displacement relations, you can reconstruct a stress field. And I'm going to skip the details of this method because I want to get to the second part. But what we see in using this nonlinear stress inference microscopy that we've invented is that indeed, if you look at the 1/r² rope network decay of forces, it corresponds extremely well to the stress decay around live cells in a variety of different fiber networks. So it looks like, indeed, there is long-range force transmission. These cells are able to stiffen and contract the network at a large distance. And that is something that they might use to mechanically communicate and send mechanical cues together to each other. A big open question in that context is, what do the cells feel? Because they're exerting strong forces, so they're going to feel the response to their own forces rather than the linear properties of the material. How much time do I have? Yes. Okay.
So it's one question, but maybe it's two. So when you say 1/r² and 1/r³, can you remind what multiplies 1/r² and 1/r³, just so that the dimensions are okay? So
you have, let's work in continuum elasticity. You have local active units that exert a stress sigma 0 at its boundary.
Mm-hmm.
And you want to know sigma of r given sigma naught, sigma 0. And what I say is that it's going to be, and it also has a size r0. Okay? Mm-hmm. The stress transmission predicted by linear elasticity is sigma of r is r/r0 to the -3 times sigma 0. Okay?
Mm-hmm.
My prediction is that if there is buckling in its network, so if the transmission is rope-like, then you will get r to the over r0 to the -2 times sigma 0.
And is there
No, because that's, I define this as the stress exerted at the boundary of the this is just to match the sigma 0 and r0 here match the boundary conditions.
What you're assuming that's just r over naught. I'm trying to figure out if there's some other factor that won't be playing there.
So far, I've assumed that inside the active unit, there's the same material as outside. That's one of the assumptions in order to get this.
And is there an effective field theory description when you are at large distances that you can assume this to be some kind of defects and then to have some long distance physics that would explain these powers?
That's just, so this is linear elasticity, this one, meaning that you're assuming that the displacement you're inducing are proportional to the stresses. So the strains you're inducing are proportional to the stresses.
Uh-huh.
Elasticity is always kind of long range. These are always power laws and not exponentials. So
I'm asking if there is a theory in terms of some field that describes the tension locally that gives the second
So,
power, the -2 power.
What you are assuming here is the first one, you're totally rigid. The second one, you're totally floppy.
I'm linear. I'm not totally rigid. I'm linear, meaning that the response in compression and in tension is the same. That's all I'm assuming.
Yes, but you're responding linearly to the system,
Yes.
right? The other one, you assume you're totally floppy.
Yes.
Is there nothing in between?
There's something in between, which is that you're going to be floppy until you're not. So if there's so typically the force displacement curves.
No, I will say, you're saying there's a threshold for buckling, and then
Yes.
after buckling, you're all floppy. That's what you're assuming. But you could imagine that the medium is not that well behaved, right?
Yes, I understand. What I'm assuming for this is that tension, compression is negligible with respect to tension. So any kind of tension stiffening, for instance, will also do the trick. If there's a finite buckling force, for instance, if that is the force displacement relationship, force extension relationship, then you're going to have losses. There's going to be an offset to this.
That sounds fine. When you look at real cells and you talk about wound healing, do people have experiments on epithelial mesenchymal? If you change when you become mesenchymal, that's why you have wound healing, that you really have a lot of networks in there? People have numbers for that?
I'm sure some people have numbers. Yeah, I can't comment on that. That said, I think you're talking about the later stages of wound healing, where there's an actual conjunctive tissue.
No, when you're epithelial, you don't have these networks.
Yes, exactly.
They're not motile, so they have to become mesenchymal. That's when the networks
Exactly.
start to build.
Yes. So
Right, so
here I'm talking about the
mesenchymal limit.
yes. So if there are no more questions, I'd like to move to the second part of my talk.
Sorry, I'm still a little bit.
Okay.
I'm a bit confused because I thought you were. Can you just quickly remind why it is -2 the power? Did you explain, or you are just saying?
I can explain again.
Sorry, thanks.
This is the cartoon that explains it.
Okay, I have a question. The structure that you showed reminds me of the so-called Voronoi construction of the random crystal. Is that related to that?
Okay, I'm sorry, I didn't hear the keyword. What does it remind you of?
So you have the structure that you showed with something surrounding each stress point, which looked like a pentagon or something like that. Okay.
Yes.
In one of your
That one.
transparencies.
So,
Hello?
yes.
Yeah, so that's sort of reminiscent of what in a disordered crystal is usually called the Voronoi construction, that defines some generalized notion of a unit cell in that case. Is this similar to that? So how are these regions defined?
Here,
Because
this
there,
region,
yeah.
this region
Yeah.
I just drew the Voronoi cell around.
There's no bond conservation here, so.
There's
Right.
no- Yes. Here I've just made a cartoon of the Voronoi cell around each of these. This is a phenomenological law for the force transmission, which is very well verified in simulations. It's
Yeah.
not exact yet. We have an exact theory, but for simplified geometry. I guess the shape of these is kind of emergent from the force balance and from the fact that, and from the initial distribution, as Ricardo mentioned earlier. So I wouldn't read too much into this specific cartoon.
Okay.
There's surely nothing crystalline that I've put in there.
No, but I don't mean that there was a crystal. It looks more like a disordered crystal in that sense. your picture is reminiscent of a disorder, of a randomly pinned crystal or something like that with vacancy. With Javi, okay.
Maybe.
So is there a reason for this? You said it depends on the Force law, but is there an optimal distribution in that sense?
I'm sorry, I didn't hear that.
They are an optimal equilibrium distribution.
So here, what I'm showing here is the static, so the equilibrium static. They're generating a constant force or a constant stress at their boundary, and I'm looking at the mechanical equilibrium solution to the force transmission. The non-equilibrium
For each region
is a completely different and much more complex set of problems.
Okay.
Just maybe he's confused. The system is run by motors, so it's very different. It's a very active system, right? So
it's an active system for which I make a purely mechanical description, which is not that active. But this is a prediction for the active stress.
This just that's the equilibrium of forces that you're doing here.
Why couldn't these exponents be something in between 2 and 3? Because a priori, isn't it a bit like these exponents in polymer physics that are complicated? And it's very rare that we can compute it exactly.
That's because mechanics is simpler than polymers, which have all the thermal effects. So here, so you would get something between 2 and 3 if you had a different slope. So this is the force extension relation for an individual filament. You would get an exponent between 2 and 3 if you had something that is kinked here. So that has a finite compression rigidity, but that is smaller than the compression, the tension rigidity. And there are a few papers on that. But there's no real evidence that there is a finite compression rigidity. So if you make a realistic model of an individual filament, the rigidity goes to 0 on saturates and a large compression, meaning that this slope goes to 0. The force goes to a constant force that we can call the buckling force. And in that case, it's really 2, the exponent. It's an exact exponent. If that's okay with you, I'd like to move on to the second part of my talk.
Sorry, let me just ask, how many of these results are new in the sense that engineers did not know them, or?
What engineers knew is this result. That's an engineering result. And the fact that for very special circumstances in linear homogeneous materials, so that's with fixed boundaries, this term vanished. I found one reference which knew that. All the rest is new. But yeah, these are questions. Okay, maybe I can comment on that because I did not expect to find such simple results that would be new at the same time. The thing is, engineers are generally interested in the effect of boundary forces onto the deformation of material and the stress, while here we're really interested in the force generated from the inside. There's some work by Eshelby and others about defects in materials, which act as effectively locally generating forces, but they didn't really look at the macroscopic stress that results from it. So that's how I found the niche of finding new results in this simple elasticity theory. Oops. Spoiler, sorry. Okay, let me move on to the second part, which is something that, so this first part was work that I'd done during the first part of my postdoc and my PhD. Now I'd like to move on to stuff that I've done more recently, which has to do with inferring force field, reconstructing force fields from experimental observations of Brownian dynamics. So this is a collaboration that I've done with another postdoc at Princeton, Anna Frischman, who now moved to be a faculty at the Technion in Israel. And what we're trying to do is, given experimental observations, learn as much as we can from about the underlying stochastic dynamical model. So to motivate this problem a bit, let me give you a very brief introduction of Brownian motion. So Brownian motion was predicted about 2 centuries ago and observed. These are trajectories that are more than 100 years old. And they result from the stochastic bumps of solvent molecule on a large colloidal object. And that gives them their erratic, non-differentiable motion. It's omnipresent inside the cell in life sciences, for proteins, for viruses, for the motion of cilia and other biopolymers. And it's also extremely commonly used as an effective model, for instance, for the motion of cells. So an effective model where it's not really thermal collisions that generate the Brownian noise, but it's more like the internal dynamics of a cell, the fluctuations that lead to El Nino dynamics in climate dynamics, the random drift of a population in population dynamics, or the stochastic evolution of a financial market. So in all of these problems, you have a dynamical system, which I'm going to model as a vector x that you observe in time, x of t, and that can be high dimensional. That's a 6-dimensional one, for instance. And the dynamics can be split in 2 parts. So you can write dx over dt, the equation of Brownian dynamics, as a sum of a force or drift, but that's the deterministic part of the dynamics, and a random noise that I'm going to model as a Gaussian white noise. So the random noise models the interaction with fast, non-observed degrees of freedom, while the force models the interaction with slow and deterministic, models slow deterministic trends. I'm going to make a key assumption here when studying this problem, which is that the force on the diffusion field depends not explicitly on the time, but only through the state of the system. So there are fields, f of x and d of x. And the question I would like to address is the inverse problem question. If you start with experimental data, an experimental trace, x of t, with a finite number of time points, how do you reconstruct the dynamical equation that generated it? How do you fit this data with the best possible model within the realm of Brownian dynamics? So this is an important problem. I've told you that Brownian dynamics is omnipresent, but why is it important to learn it from data? Well, if you look at climate dynamics, it's absolutely crucial to use it to make predictive modeling and look at the statistics of the future evolution of climate. For cell motility, for instance, it's really useful to correlate the behavior of a cell with its phenotype and genotype. If you look at a particle tracked with an instrument such as an optical tweezer, a laser that tracks particle, it's going to be important to calibrate it and to control the force fields that you exert to manipulate soft and biological matter. In a number of cases, you cannot measure the interactions just using the laws of thermodynamics because of the out-of-equilibrium nature of many active systems. So it would be a means to measure the interactions. If you look at intracellular traces of single molecules, it's going to help, analyzing them is going to help you understand the complex environment of the cell inside the cytoplasm and the nucleus. And finally, if you look at a single molecule randomly moving along a filament, you can use it, for instance, to quantify dissipation, try to extract energy from molecular level machines and such objects. So this is a very broad and important question. Now, why hasn't it been all solved before? Why is it a difficult problem? Well, the goal here is to reconstruct the force on the diffusion field given data. And it's clear that if you have a very short trajectory, such as this one, not much is going to be possible. While if you have a long one, such as this complex three-dimensional attractor, maybe there's more information and maybe you're going to be able to do more. So there's a conceptual problem here, which is how much information is present in the data. And then there's a second, more practical question, which is, even if there is a lot of information, how do we in practice?
Let me ask a simple question, just a simple question here. You are looking at a single cell, or you're looking at multiple cells and the same cell on different ranges? How you do this sort of machine learning here? I'm trying to figure this out.
So now I'm looking not specifically at cells, but at the general question of learning force on diffusion field.
Right, but if you repeat this experiment many times, you're going to get different trajectories, right? So do you have multiple trajectories so you can go back to your equation, or you try to get it out of a single trajectory?
So my goal here is to get as much as you can from a single trajectory in a statistically controlled way.
Okay, let's see where you go.
So there's a number of, so there's a question of how do we do in practice, and then there's a number of other challenges, which is the fact that it's not a simple filtering problem. It's the noise actually shapes the trajectory and changes its properties. So it's hard to disentangle from the drift, from the deterministic forces. Then you can look at high-dimensional problems. For instance, trying to learn the interaction between cells from the motion of many cells at the same time. You have limited data. I'm going to try to do as much as we can from a single trajectory. Your problem is out of equilibrium, meaning that your standard thermodynamics approach won't work. And typically, because we're looking at data that's obtained with a microscope or with a measurement instrument, you have imperfect data. You have a noise on a measurement noise on your data that is going to add a tremendous challenge to the inference problem, and that I want to take into account too. Okay, so let me go a bit deeper into the structure of this problem. So let's now look at so this is the Langevin formulation of this Brownian dynamics problem. I'm going to transform it now to the Fokker-Planck equation, in which I'm going to write the dynamics of the probability density, P of xt, as a function of time. So it's equal to minus the divergence of the currents, of the fluxes. And these currents can be written as the difference between two terms. Drift part, the force times the probability. I'm sorry, I'm using drift in the opposite way as population dynamics people do. For me, it's the force. So a force part, which is F times P, minus diffusive part, which is D grad P. And if you look at the steady state solution of this system, so I'm going to look at a system that has relaxed and that is at steady state in its PDF, then the force can be split into two terms by just looking at using this decomposition and saying that this is 0. The force equals the velocity plus confinement term. So the velocity is an active out of equilibrium term that it would be 0 at thermodynamic equilibrium, and it's equal to the flux divided by the probability density, J over P.
D is not constant.
I'm not assuming that D is constant here. I'm going to.
D appears inside the.
That changes the definition of the force. It's. I'm going to make the to take this definition here. That's a discussion we can have later if you want. So there's the velocity, and there's a second term, d grad log p. So I've divided by p in this first part, and that part is essentially at equilibrium, and it just confines the system to stay where it is. So I can make this decomposition explicit using tracks of a simple system that I've simulated. So this is a simulated trajectory of an Ornstein-Uhlenbeck system, meaning that the force is a linear function of space. And here I've made it out of equilibrium, so you see that it circulates at the same time as it confines the system. And if I make this decomposition, I get the rotating part, which is the force, the velocity, which is active, and the confinement part, which is central. And in the past few years, there's been really a lot of interest in trying to quantify these active currents and connect them to dissipation, in particular inside cells, which was to a large extent sparked by this paper published in Science 4 years ago, in which they looked at the fluctuations of a single filament inside a live cell. And if they plotted the time traces of the angle it makes with the base and the curvature of the filament, they get this random trace. And putting in the phase space of force and curvature, you see that it rotates. So there's a very high noise, but there's a statistically significant signal that shows that it rotates, and there's an active, irreversible dissipation in this system that this paper put the focus on. And I think this paper brought many people, including myself, to look into this field because we thought it was really interesting, and the way they were doing it was terrible. They just coarse-grained, so binned the trajectory and counted transitions between one state to the next. And this is a pretty inefficient data analysis technique. So I'm going to try to, as a byproduct of the force inference problem, I'm going to try to reconstruct these currents and to use them to infer what is called the entropy production rate, which I'm not going to talk too much about, but it quantifies dissipation and how much out of equilibrium your system is. Now, moving back to the force inference problem, let's look at the problem of force inference in more detail. So if I discretize over time delta t.
Sorry, I have a stupid question again. Just vectors and numbers. D is a vector, or what is?
D is a tensor. It's a matrix.
Ah, okay.
X and F are vectors.
Okay, thanks.
Yeah. So if you discretize over time delta t the Langevin equation, you get that delta x is the force times delta t plus a term that is square root 2d times a random variable, random normed Gaussian variable, times square root delta t.
And you assume that's enough. Basically, you're going to throw all this left flight into your force somehow.
Yes. Yeah.
Do you know the dimension of X from data?
Okay, so that's a great question. The dimension of the data you take is known. It's what you measure, right? The dimension of the system, the intrinsic dimension of the system which obeys this equation can be different and can be significantly larger. I'm going to assume either that you measure all the variables that enter here, or that you want to look on averages over the hidden variable. So everything I'm going to say here are true in terms of being averages over the hidden variables that would evolve. And that would include some terms of some colored noise effects, for instance, in which there's a memory going on. Okay, so let's evaluate the difficulty of the problem. There's two terms that we want to infer, F and D. Now, from this equation, at a single time step, I can make an estimator for each of them. For the diffusion coefficient, you just take delta x squared over 2 delta t, and that's the diffusion tensor, plus something of order 1. Because this is higher order, and the noise coming from this is of the same order as the square of this variable. So this is essentially a low noise problem. Each data point you get brings you something like 1 bit of information about your diffusion tensor. And typically it's not regarded as being a hard problem to reconstruct the diffusion field. On the other hand, if you look at the force estimator, well, you can get 1, 2. Delta x over delta t is the force if you condition on the initial point, so that this term vanishes on average. But the correction for a single data point, the correction to this is of order delta t to the minus 1/2, and so that's a divergent correction at small times. Well, if the times are long, of course, you're not resolving the force because it varies in time. So this is a much harder problem. It's a high noise problem. And for this reason, I'm going to focus on it now. I'm going to assume that the diffusion is constant and known for the rest of the talk. That's in the paper that we wrote. We also propose a method to infer the diffusion, and don't assume that it's space independent. So I'm going to tell you only part of the story here. But then, then we're going to focus on the force field, and you really have there's a bottleneck of information here, meaning that you need to filter it through this high noise problem.
Sorry, I have a question. this local diffusion constant in principle can be measured, right?
Say again?
The diffusion constant, the local diffusion constant, in principle can be measured.
Yes. It's a much easier problem than measuring the force.
Right. So it's not, why can't you just get that from the data, from experiment?
Well, you can measure it to some accuracy.
Yes.
And so here I'm neglecting the fact that the estimate is inaccurate, and I'm going to assume that it's known.
Okay.
Well, you're going to do some short time velocity loss correlation function. You're going to get your D from there, right?
Yes.
Yeah.
But then if it has a complex space dependence, then it's a bit more complicated. And that's a problem.
Short time is good enough.
Yes.
It's actually in ecology, it's like a super open problem estimating. Even estimating D from GPS location data of animals, for instance, in ecology, is an open and hard problem. So.
Yes, because you'll have to figure out what's your time interval you're happy with. But he's assuming that's not the hard problem. He's making an assumption here. Let's see where he goes.
Yes. The method is geared towards soft matter and cell biology data so far. But I'd be very happy to discuss possible applications to other fields. OK, so I was saying that this is a high noise problem, measuring the force. And so and I'd like to show that actually information comes at a finite rate in this problem. So to do that, I'm going to make an analogy between this this Brownian dynamics problem and the communication theory problem. If you look at this individual time increments, you have a signal part, this f delta t, and a noise part, the square root 2d xi, and you want to extract the signal part. So it's a bit like having a communication channel in which the emitter, the force field, sends a signal, but the physical system receives that signal only in a noisy version in which you've added the Brownian noise. And that's not just a superficial analogy. Actually, you can quantify the rate at which one can extract information from such a communication channel. It's called the Shannon-Hartley capacity.
That's the small noise limit, right?
Say again?
That's the small noise limit.
No, it's
general.
Yeah, that is general. Yes, it's an upper bound to the information you can extract, but that upper bound is general. That upper bound says that the capacity of the channel, so the number of bits of information per second that you can extract by monitoring such a communication channel, is equal to 1 over twice the time interval, 1 over 2 delta t, times the log of 1 plus p over n, where p is the signal power, the mean squared signal, and n is the mean squared noise. So here the signal was f delta t and the noise was this noise term. So if I put the mean of each of them, it gives me a capacity that is 1 over 2 delta t times log of 1 plus f squared delta t squared over 2d delta t. The delta t simplify here, so it's log of 1 plus something of order delta t. So in the small delta t limit, this converges to a finite rate, because the delta t simplifies here again. And so the limit when delta t goes to 0 is f squared over 4d. So this is the maximum rate at which you can extract information from from monitoring Brownian dynamics, extract information about the force field. And this is a new result. I've derived it in a somewhat hand-wavy way here. The rigorous version of the capacity of this system is 1/4 of the average, so a long-time average or an average over the steady-state probability distribution of f dot d minus 1 dot f, where here now this is true in tensor way. So this is a time scale. This is an inverse time scale, which can be measured in bits per second if you multiply by log 2. You can write it as a rate too, the information rate, so it's the limit of the average limit of the information divided by the time, where now I've defined the information as the log of the ratio of two likelihoods, the likelihood of the observed data, of the observed trajectory in the force field, divided by the likelihood of the data in the absence of any forces. So what it means is that this capacity quantifies the rate at which your system becomes distinguishable from pure Brownian motion, the rate at which you have a structure that emerges from from the trajectory. And so this information is a well-defined quantity, and I can put a number on the previous trajectories that I've shown here. This trajectory has 12 bits of information, while this one has 5 kilobits. And so there's clearly much more information here than here, and so you're going to be able to do more about inferring the force field. This capacity actually has a very nice relation to the stochastic thermodynamics quantities that people are used to. If you get back to the decomposition of the force in terms of a velocity and a confinement part, so that was this decomposition of the rotating force field in terms of a pure rotation and radial force, and you plug this decomposition into the formula for the capacity, f d-1f, so this is quadratic in f, what you get is that the cross term between velocity and diffusion and confinement vanishes exactly. And so 4 times the capacity is a sum of 2 positive terms, which are quadratic forms in each of these components. The first one is the entropy production rate. It measures how fast you're dissipating and how fast you become irreversible. While the second part is what is called the inflow rate, and it's on average minus the divergence of the force field. It quantifies how much convergent the force field is and how it confines you. So in a sense, you have a finite rate of information in your system, the capacity, which decomposes into 2 types of orders. The first one, which is dissipative, which is irreversible, which tells the system where to go. It's forward in time, and if you reverse the trajectory, you would go the other way. The other part is time reversible. It's a confinement term, and it tells the system where to stay. It basically fights against the diffusion forces that want to send your system away. And these 2 together from the capacity, which is the rate at which you can extract information from your trajectory. Now, this is nice and fine, but these were like theoretical considerations. How do we exploit this information in practice? How do we use it to reconstruct the force field? And as a bonus, to decompose it into this reversible and irreversible motion. So that's going to be what I'm going to focus on.
Right, we're assuming that that's the answer, that's on that format, right? You don't have a curl term that you have to have an orbit. You're assuming that that's sufficient in there.
The velocity contains the curl term. Basically, the velocity is the current divided by the steady state PDF, and the current is per pure curl at steady state.
So the information, you're assuming it goes with time, is that right?
Yes, it goes linearly with time.
Is that an assumption?
It's a result from this slide, essentially. Each data point adds finite information. I want to add that it's an upper bound on the total. So the capacity gives you an upper bound on the total information. So defined this way, this quantity goes linearly with time.
You get some closed cycle and you stop getting the information.
You're never going to get perfect information about the force because it's a noisy dynamics problem. And so each time you go even if you go an infinite number of times to the same point, you can always mix up the effect of Brownian noise with the effect of the force. So you're never going to be sure that there's a force in this direction. Did I answer your question? Okay, so now I'd like to ask how we can use this information in practice to reconstruct force and diffusion, the force field. And let's start with a simple example, the case of inferring a constant force. So it's not very exciting, but it's going to be instructive. The best estimator for a constant force, for instance, for this trajectory, is going to be the end-to-end distance divided by the total time. So delta x over tau. The interesting part about this example is that you can also self-consistently put a confidence interval over this result. by assuming that the fluctuations are Gaussian and that the noise is Gaussian. And that confidence interval has a nice form, which is the square root of the space dimension. So that's the number of variables you want to infer. That's the number of degrees of freedom there is in this constant force, divided by twice the information that I just defined. So the information being the capacity times the total time. So here, for instance, there are 9 bits of information and 2 dimensions, and that results in a 30% confidence interval around this inferred force. And indeed, you would get that the total force falls within this circle. So that's what limits inference for the case of constant force, this ratio between number of degrees of freedom and total information. Now, if we want to learn a complex force field, a force field that varies in space, then you're faced with a problem, because you have a finite total information that I just defined, but an infinite number of degrees of freedom, the force value at every point in space. And so you're going to need to do some dimensional reduction in order for this inference problem to be possible. So one way many people proceed is to do grid binning, which works in 1, 2D, perhaps 3D, but definitely not above that. You just put your data on a grid, count transitions, and assume that the force is constant in each of these bins. There are many reasons why this is not a great way to do it, and I'm going to propose a different method. There's also a number of different phenomenological ways of getting the force field that are generally not so principled. I'm going to focus on a new method that I've introduced with my colleague, which we call stochastic force inference. And the idea is very simple. It's performing a linear regression of the force field with a finite set of state-dependent functions that I'm going to take to be smooth, B alpha of x. And so I'm going to fit the force field, f of x, as a linear combination where these f alphas are inferred coefficient of known functions b alphas. I'm going to use two principles to infer these fitting coefficients. The first one is that I want them to be maximum likelihood estimators. But then I'm going to, that's not enough, because that would make them extremely sensitive to measurement noise. I'm going to add a second trick, which is to modify this estimator using the so-called Ito to Stratonovich correspondence, which makes them robust against measurement noise, as I will show in a minute. I'm not going to go through derivation, but I have a rigorous proof of convergence of these estimators. And I'm going to show you only the final result, which is exactly what I put in the code to infer force fields. So my input is a discrete time series, x of time 0 till x of final time. I'm going to start by selecting fitting functions to fit the force fields. And that's where having an educated guess on the form of the force field would help. But if you don't have an educated guess, you can just use a generic basis, such as polynomials or Fourier modes or wavelets. So that's my B alphas. A technical step is that I'm normalizing them by their covariance to get these normalized projectors C alphas. Then this is the big important step. I'm going to infer the maximum likelihood coefficients. These F alphas coefficients, which are constants, are going to be the time average of the difference of two terms, dx over dt times C alpha of x, where C alpha is evaluated at the midpoint of two intervals of this time interval. That's called the Stratonovich convention for stochastic calculus, and that's very important here. Minus d times the gradient of the fitting function, and that's where it's really important to have smooth fitting functions, because I need to be able to take their gradient. That's all. This is the exact formula for the maximum likelihood estimator. I also have convergence rates with which I can self-consistently estimate the relative error on these fitting coefficients. And this relative error is given by the square root of the number of fitting functions times the space dimension. So that's the number of fit parameters in total, divided by twice the inferred information. And so that's where the inferred information is available from the data. So that connects it to the constant force case. It's really the ratio between the number of degrees of freedom to the total information available that controls the precision of force inference. Using this estimate of the error, you can adapt the basis to optimize over both underfitting, so not good enough representation of your data, and overfitting, meaning starting to fit the noise and take it as signal. And there's a self-consistent way to loop and to adapt the basis size. All in all, if you end up with a smaller than one relative error, you've resolved some signal, and you can reconstruct a force field as the sum over all fitting functions of the fitting coefficients times the fit function. That's all there is to it. There's no black magic or deep learning networks.
I have a couple of questions here. This looks very applied math. Do I learn any physics from it? Unless you tell me what the C alphas are and they have some intuition here?
So it's applied math, if you want to call it like that. It's the best way to fit a force field given the data on a set of fit functions.
Is this like the maximum entropy method?
No.
It's a maximum likelihood. It's among all the functional forms as a linear combination of the fitting functions is the one that explains the best the data. One physical thing that you understand, that you get from here, is that actually there are two terms here in this sum, and they correspond one-to-one to the velocity and to the confinement terms. So you can disentangle active forces that produce entropy, so you can compute entropy production using this, with an error bar to how much entropy was total produced, and the confinement part.
Is there some similar methods? Basically, people haven't tried to do something like that before. They probably did, right?
So I think we're the first one to use the Stratonovich fitting here. There's been some one-dimensional maximum likelihood fitting.
But in G networks, people always have been trying to break it velocity and confinement, right? So I don't know if there's similar methods that...
Not that I know of.
Okay.
Yeah, we've been through a lot of papers trying to solve that same problem. Very few had results that were convincingly convergent, meaning that in the limit of a long enough trajectory, you would actually get an ideal representation of the force field. Many were heuristics that give a non-controlled approximation. And then among the ones that converged, it was always one-dimensional, and they did not consider measurement noise. So let me show you. So yeah, if you want to try it with your data, it's on GitHub. Let me show you how it works on a couple of examples. The first one is a linear confinement term. Actually, it's the same trajectory that I showed before. If I fit it with an adapted basis, meaning with also a linear form, this is the force field that I would get in blue. I could also infer the information, estimate the information, 40 bits. That gives, there's 6 degrees of freedom, 2 dimensions times 3 function, 1, x, and y. And so that gives me a 30% confidence interval for the force inference. And indeed, this is the exact force field that I use to generate the data. And it's within 30% of the inferred force field. Now here I'm cheating quite a bit because I'm extrapolating the force field outside of the range where there is data, which is something that you can do if the functional form is the same for the data and for the initial model, but not in general. So really what you should be looking at is how good you're doing along the trajectory. And this is a plot of the inferred force components as a function of the exact force components along all points in the trajectory. And you see that they align closely with this black equality line between the two with a very small error on average.
But here you have a reasonably simple force field, right?
Yes. How about we move on to a slightly different one, a slightly more complicated one. So that's now the stochastic Lorentz process, which is a 3D out of equilibrium complicated force field. It's actually polynomial, but I'm not going to assume that I know that. I'm going to try to fit it with Fourier modes. What people would generally do before would be to bin it with using maximum likelihood bin. And then you would get a mean squared error of about 15%, so 85% accuracy, using this 6 by 6 by 6 grid. If I use now an order 2 Fourier mode, Fourier basis, so Fourier functions that depends on each 3 components and 2 wave vectors of cos and sine, I'm going to get 95% precision. So a significant increase in the precision of the method. It sounds a bit incremental, but if you look at the convergence rate of the 2 methods, so binning in blue and stochastic force inference in orange. Now if I plot it in log-log, you see that the blue method...
Something has a global behavior, you're going to do better. There's no way out.
Yes, yes. It's not just the fact that it's global, it's the fact that it's smooth, and that smooth functions are more dense into the space of smooth functions than binning. So that there's a better representation that increases the rate of convergence. Because essentially the force fields that we want to infer in general are smooth as a function of their variables. So that was with perfect data. Now let's add a bit of complexity and see what would happen if you added a bit of measurement noise, because all experimental setups have a degree of noise that come with them, and that's going to blur the data. For instance, if I add this point spread function, which just complicates a little bit the force field. Now, any method that relies on Ito fitting or initial maximum likelihood will completely decorrelate and go to more than 50% mean squared error, because there are dangerous biases that come from correlation between a fitting function, which can be a bin, and the position at which you evaluate the functions. So that there's a bias that comes from these two that goes as the error squared divided by the time interval. So that diverges when you have a small time interval, and that biases dramatically the signal, the inference. Now, what we've proposed is to use Stratonovich fitting, which is a modification of this, which now, if I add the same level of noise as before, so that's where the noise limit was before. Now the force inference has absolutely not budged. It's as good as it was before, even though each of these time steps is pretty blurry. If I crank up the noise by a factor 10, so now each of these butterfly wings almost start to be blurry, you still retain a lot of precision, 90%, 80%. And you really need to go to crazy level of noise like this to go to the same level of error as before. So this is the standard noise limit using usual maximum likelihood techniques. That's where you lose signal. This is where stochastic force inference will lose signal. If you make it on a plot, usual techniques fail at a noise amplitude that is of order the force times delta t. Our fails when the noise amplitude is larger than the spatial structure of the force field. So when the butterfly wings become blurry. So in that sense, I think we can say that this method, stochastic force inference, is a high-resolution technique that is particularly adapted to low-resolution data, such as single-molecule tracks. I'd like to end with a last example that is even more complicated. This time I have not one, but many particles that are interacting. They're self-propelled. They're bumping into each other. What I want to reconstruct is the interaction between them, the way they align, the way they interact. And if you were to do that, the brute force, using, for instance, Fourier modes to represent this, there's no way you would get the interaction between them, because there's 25 particles here, 3 degrees of freedom for each, the orientation and two positional degrees of freedom. So that's a 75 degrees of freedom, 75 dimensional phase space. Nothing can be done with that. However, if you treat these particles as identical, you can use the symmetries of this force field and make an ansatz that is invariant under exchange of particles. So write the force on particle I as a one body point, as a 1-point force, a 2-point force, possibly 3-point forces, and now try to fit the coefficients of these forces in a way that does not depend on the identity of the particle. And if you do that over a trajectory of 10,000 frames, you get this kind of representation of the instantaneous force field, which agrees extremely well with the initial data, with the true force field in black. If you look at the inferred versus exact force, the dotted line are the margin of errors predicted by force inference, and all points fall at, within less than 1% of error. So that's with 10,000 frames of the previous movie, meaning 5 minutes approximately. Now, if you have a biologically sensitive system that will evolve in time or that will bleach because the fluorescent proteins can have a finite lifetime, you have less data, and so for instance, this is what you would get with 25 frames, 1 second of the previous movie. You already get about 90% precision of the force inference, and definitely a good representation of the structure of the force field. So that's it. What I've introduced to you is a new way to infer, to analyze stochastic trajectories and infer the underlying model. There's 2 key insights that we've used. One is using smooth functions that utilize the symmetries of the force field. The other part is stochastic integration, a Stratonovich integration, which filters out the measurement noise. It's nice, it's on GitHub, it was accepted last week in PRX, so it should appear soon. And so I'd like to say that for me this is a nice method, but I'm not a mathematician. What I would like to do now is to apply it, because this is a means for me. It's not an end in itself, the designing inference methods. So I've told you about a few motivations of systems in which force inference is important. I've actually started working with a number of experimental groups to try to infer force fields from their data, from driven colloids that you can drive using optical tweezers, raw movies of intracellular structures, fluctuating membranes in which you want to infer the field theory describing it just using the data, colloids that move along chemical gradients, individual genes inside the cell nucleus, or microtubules that fluctuate inside a cell. And that's just an open list. There's really so many different fields in which the stochastic dynamics is used as a model. Your data could be here if you do experiments. And with that, I would like to thank you for your attention.
More questions? So all your experiments are related to biology, but why? It's just, I mean, it seems that the things you describe, stochastic processes, not from, well, at least naively, it's not necessarily biology. Is there something I'm missing? Or?
There's a large number of biological problems, biological systems that undergo stochastic processes. It's a part of the, it's only a part of the applications. Here, microtubules, genomic domains, these are directly biological applications. I'm trying to understand, for instance, the way genes interact with each other, attract or repel. So that's a very hot problem in this field. But for instance, driven colloids or chemotactic colloids, that has nothing to do with biology. These are soft matter problems.
Where is the active part in all of this? I fail to see that.
What is the?
Where is the active part of this? In principle, this looks to me like a system with a lot of internal noise.
So
you're trying to find the best fit to determine the forces. I don't see anything that is particularly active here.
So here, each of these particles, you see that they have a big white circle on a black dot. The black dot pushes on the particle. And that's, this movie is made to mimic experiments done by the group of Jeremy Palacci at UC San Diego, which has particles in which the black part is made of platinum. And there's a chemical reaction at only that part that propels the particle forward. So that's something that I've put into the model when I made the simulation.
Well, that's the point is trying to make that your active part is going to come into your flow part of the problem, but you're not going to get the origin of it, right? Basically,
You're not going to get the origin of it.
That's right.
This is
Exactly.
this
No problem. Just
yeah.
trying to say where he's getting.
It's what my method is going to tell you is that this part is active. It dissipates because it's time irreversible. If you play the movie in reverse, that part of the force field would reverse. It's not going to tell you what the origin of it is because it's not in the data.
So what are the implications of this for biology? I mean, why should the biologists be interested in this analysis?
Good question. So I've told you about a couple of applications. So already I've discussed a bit the interaction between genomic domains, which is something that I'd like to do now. Generally,
yes.
you will understand the interaction between the large objects in the cytoskeleton and the active fluctuations inside it, for instance. That's one possible application. You can use it to quantify dissipation and say how much energy is injected actively by a cell into doing a specific process. That would be an activation. That would be an application. Another one at a slightly larger scale is that I'd like to use this type of methods to model the behavior of individual cells, of small animals, or even flocks of birds, for which I need actually to extend the method to inertial cases. And that's a paper that will appear tomorrow in the archive. And then I think this could also be applied to other types of stochastic systems in biology, such as population dynamics. And yeah, that's some. I hope to learn something during this visit about this.
Any other questions?
Thank you.
Okay,
Thanks
so
a lot.
thanks.